Okay, I think we can go ahead and get started. Hello and welcome to the Joint Invasive Mussel Collaborative and Great Lakes HABs Collaborative Webinar on the role of dreissenid mussels in nutrient cycling. This topic has been of significant interest lately following a recent report from the International Joint Commission's Science Advisory Board on declining offshore productivity in the Great Lakes. The report describes unequal mixing of nutrients, in part due to invasive dreissenid mussel trapping nutrients in the lake's nearshore regions, which results in too many nutrients in nearshore areas and too few nutrients in offshore areas. The report makes recommendations to improve coordination between water quality and fishery managers and increase our understanding of ecosystem dynamics and nutrients in both nearshore and offshore waters of the Great Lakes. My name is Sam Tank, and I am joined by Ken Gibbons with the Great Lakes Commission. On behalf of the Invasive Mussel Collaborative and HABs Collaborative, we want to thank you for participating in today's webinar. Before we get started, we just wanted to provide a bit of background on each collaborative. The Invasive Mussel Collaborative was convened in 2015 to identify opportunities to advance scientifically sound technology for the management and control of invasive dreissenid mussels in order to produce measurable ecological and economic benefits. The Invasive Mussel Collaborative published a management strategy in 2018 to reduce invasive mussels and their negative impacts. This strategy identified restoring balanced productivity in impacted lake systems as one of the 5 goals and set develop and apply models to evaluate and test current assumptions regarding zebra and quagga mussel ecosystem interactions as one of the objectives of that goal, which is consistent with the Science Advisory Board's report. The Great Lakes HABs Collaborative was formed with a mission to improve communication among scientists and between scientists and decision makers on issues related to harmful algal blooms in the Great Lakes. The HABs Collaborative is helping to establish a common knowledge base of where science currently is on harmful algal blooms and what future science may be needed, and how the region can work together to prevent and manage HABs. Today's webinar is part of the Invasive Mussel Collaborative Network, facilitated by the collaborative itself and the HABs Collaborative jointly. The purpose of these webinars is to facilitate learning and information sharing on topics of common interest within the invasive mussel community. This webinar will be recorded and archived along with past webinars and future webinars to come on our website. If you'd like to learn more about the collaborative or interested in staying in touch with our communication network, we encourage you to visit our various websites or join our listserv. Today's webinar will cover the role of dreissenid mussels in nutrient cycling. We are so pleased to have our 2 speakers joining us today to discuss these topics. While we always strive to minimize technology issues during these webinars, we know that issues may arise, and we do apologize for those in advance. But I'd like to cover just a few logistical items before we get to the presentations, just to make things run smoothly. Audio for this webinar is automatically going to be streamed through your computer speakers, unless you choose to call in using the connection information sent out in the registration confirmation email. In order to minimize any potential for interruptions and background noise, we've automatically muted all attendees, and attendees will remain muted throughout the webinar. You do have the ability to start your video. However, to save on bandwidth, we do ask that all attendees turn off their cameras. We do encourage and accept all written questions from attendees during the webinar. So if you have a question, you can type that right into the chat box, which is signified by the little chat icon, and that's circled in green on the screen. Questions can be submitted anytime during the webinar. However, we are going to answer questions after both presenters have given us their presentations. All question and answers will be facilitated at the end. We'll do our best to take questions and comments during the webinar, but if you do need to leave before we've gotten to your question, we do encourage you to review the webinar recording that will be posted on the website so that you can hear your answer. If you're experiencing any technical difficulties, you can send a message through the chat box, and you can direct that message to Ken Gibbons. So without further ado, I'd like to introduce our first presenter, Sergei Katsev. Sergei is a professor at the Large Lakes Observatory for the University of Minnesota-Duluth. After receiving his PhD in physics from the University of Ottawa in 2002, he worked on developing models of sediment geochemistry in marine and freshwater environments. His current research interests include sediment geochemistry in the Great Lakes, physical and biogeochemical processes in tropical lakes, and using modern lakes and biogeochemical analogs of the environments on early Earth. His recent work on categorizing the effects of quagga mussels on the geochemistry of sediments in Lake Michigan and Lake Huron was sponsored by the U.S. National Science Foundation. And I'll stop sharing, and Sergei, whenever you are ready, you can take it away.
Okay, thank you. I'll try to share the screen. Okay, and did it work?
Yep, looks good. Thank you.
Okay, thank you. Hello, everyone. Thank you for inviting me. I'll talk about the nutrient cycle in sediments of the Great Lakes, and I initially planned to cover both phosphorus and nitrogen, and I quickly realized I cannot cover both of them. So this talk will be about phosphorus. We've had a NSF-funded project to look at the deep sediments in two of the Great Lakes, Lake Michigan and Lake Huron, to characterize sediment geochemistry and the exchanges of nutrients and the roles that mussels, invasive quagga mussels, played on them. So I'll report a little bit on the findings from that research, but mostly I'll talk about the more general features of phosphorus recycling in lakes and how mussels may affect them. So if you're, okay, all right. If you're familiar with phosphorus recycling in sediments, you probably have in mind something like this picture. If not, I'll quickly go over this. You're looking at the sediment. So this is actually a picture of sediment core in Lake Superior as an example. Phosphorus is delivered to the sediment with settling organic particles. Organic particles start to decompose inside the sediment. They release phosphate. If phosphate is released close to the sediment water interface, it can diffuse back into the overlying water. For deeper mineralization, when it's released in the reduced sediment, it can diffuse upwards, and frequently it precipitates on the surfaces of iron oxides, which serve as a trap for phosphorus. And here is the typical profiles that we find in Lake Superior. Here is the SRP, soluble reactive phosphorus. It accumulates in the reduced sediment, and its concentration in the oxic sediment is fairly low, but nevertheless, it can still flux out of the sediment. So in well-oxygenated lakes, these fluxes of phosphorus back into the water column are regulated primarily by organic matter mineralization. In more productive lakes that experience anoxia, these phosphorus fluxes may be linked to the dynamics of iron. When the lake goes anoxic, iron oxides dissolve and phosphorus can flux back into the water column. So this is typical in smaller seasonally anoxic lakes. Sediments do contribute a significant portion of phosphorus inputs to the water column, even in large lakes. This is schematics that we put together for Lake Superior. So flux of phosphorus out of the sediment is comparable to the external inputs of phosphorus into the lake, depending on how you count it, between 15 and 40% of the total phosphorus inputs. The budgets for other Great Lakes are not very well understood. There are estimates in Lake Erie on the order of 20% of total phosphorus inputs come from sediments. Lake Ontario, Michigan, and Huron, I think the budget is known even less, unless I missed some papers. But the point is, sediments are significant as sources of phosphorus for the water column. And of course, phosphorus is a limited nutrient. That's what controls the primary productivity. A lesser known aspect about phosphorus cycling is that this efficiency of recycling of sediments, what percentage of phosphorus comes back from the sediment, it's an important parameter in the dynamics of the lake. If you think about the lake as a sort of single box, this simple system, phosphorus enters the lake from the watershed, and you can regulate this. We regulate phosphorus inputs. If you think about how quickly the total phosphorus concentration in the lake will respond to those inputs, you can fairly well figure out that it depends on how much phosphorus actually cycles in the lake. Phosphorus gets settled as organic particles, gets recycled in the sediment. So if the size of this loop is substantial, it takes longer for the lake to respond to changes in external inputs. So if you reduce external inputs, it takes longer for the lake to adjust. Likewise, if the lake is being flushed quickly, if the hydrological residence time is low, the lake would respond faster. So I had a paper a couple of years ago where I put this into basically mass balance equation, and you can calculate the typical time scale in which the lake responds. So for lake, I just plotted these for Lake Michigan, Lake Huron, Lake Erie. For typical recycling efficiencies there in the lake, it's about 20–25% in Lake Michigan and Huron, probably a little higher in Erie. This is a guesstimate. So the lakes respond on the time scale of several years, which is remarkable given their size. And I'll be coming back to these graphs at the end of the talk. You can do another calculation from the similar considerations and calculate what's known as internal load, as a ratio of internal to external load. This flux of phosphorus from the sediment is known as internal loading. And you can get these numbers that, again, so around 20 to 40 to 50% of total phosphorus loading comes from the sediments. But the point is that it's possible to calculate, and we can figure out how these parameters change if something changes in the lake, particularly if something changes in the sediment. This efficiency of recycling, what the percentage of phosphorus comes back from the sediment. So now we go to the quagga mussels. Most of you probably saw a version of this slide. It shows the progression of quagga mussels through Lake Michigan and Lake Huron. They reach extremely high densities, up to 10,000 individuals per square meter. I will show that these maps are not completely accurate. They show complete coverage of lake floor by quagga mussels, even in the deepest portion. This is not quite what we find, but this gives a good idea about the massive changes that happened in these lakes. This is just a slide from our data that shows the light penetration in these lakes. This is basically the intensity of light on log scale as a function of depth within the water. Lakes Huron and Lake Michigan are now about as transparent as very clear oligotrophic Lake Superior. There is still light there around 50 to 60 meters. That's quite remarkable. While Green Bay, of course, is much more turbid. Green Bay of Lake Michigan. What happened to the productivity of the lake and transparency is related to primary productivity, which is controlled by phosphorus. And what was observed throughout or over the last several decades is that the total phosphorus concentrations in the lake, the typical average phosphorus concentrations in the lake, they seem to have decoupled from the inputs of phosphorus from the watershed. So this graph, this is Glenpoe data. It shows phosphorus concentrations in Huron, Michigan Superior. Basically what it shows is that starting around year 2000, around the time that mussels invaded these lakes, The phosphorus concentrations in the three lakes, so Michigan and Huron, were decreasing towards where they are in Superior. So the levels are now very similar in all of these three lakes. If you put this in a model, this is again was done several years ago, you will find that this decrease cannot be quite accounted for by the decreases in external loading. So given the external loading that we know of, the concentrations has to be, the concentrations have to be a little higher. Nevertheless, the concentrations continue to decrease despite the external loads being about the same. So this means that something happened in the benthic system. Something happens that changed these dynamics. So we went out to characterize the sediment geochemistry. This is just a quick map of not all of the locations, but I think most of the locations that were visited. What I wanted to show is the color coding corresponds to the presence of mussels. So green dots correspond to places where with significant mussel coverage, blue dots with no mussels, and cyan is somewhere in between. Perhaps there are a few mussels there. There is not a good correlation with water depths, and in fact, we find water depths not to be a good variable. It doesn't correlate with oxygen uptakes, carbon mineralization. It's not a good predictor for many sediment characteristics. Pretty much the only characteristic that we find correlating with the presence of mussels is the sediment porosity, basically how dense the sediment is. So this graph shows the porosity of the sediment as a function of depth below the sediment surface. We find mussels in locations where this porosity drops significantly, just below a couple of centimeters. In these lakes, this corresponds to all the sediment exposed post-glacial hardened muds. If in places where softer, fine-grained sediment accumulates, so these porosities on the order of 0.9, we typically don't find the mussels. I'll show just a couple of more slides that may be illustrative for the effects of mussels, not directly relating to the main story. We found that mussels are bioturbators, so this is a time-lapse video for mussels moving around in a sediment core. We could calculate the intensities of these bioturbation. They seem to be significant, although they're typically in the top 1.5 centimeters of sediment, maybe not quite as deep as in some sediments that do not have mussels, that have more native bioturbators. But so mussels do move around the top of the sediment. Here's a typical pore water phosphorus, soluble reactive phosphorus in these cores. So the red lines are concentrations of phosphorus. On the lower scale, the black lines are concentrations of oxygen. Basically, you could see the depths of oxygen penetration in Lake Michigan, Lake Huron. Here is Green Bay. Green labels here correspond to cores that had mussels. Again, so blue, no mussels, cyan colors, something in between. These are actual pictures of sediment cores. No clear pattern associated with mussel presence here again. Here's these dense sediments, post-glacial sediments that have very low organic carbon content. They lower phosphate concentrations, but this is related to the properties of the sediment, not the presence of mussels. And like I said, this turns out not to be terribly important for the main story. I'm getting to the main story now. So what we find is that mussels actually dominate the phosphorus fluxes in and out of sediment. These are results from a number of different sediment incubations performed by several people in our group. So what you see is the phosphorus flux out of the sediment cores, or out of the sediment, as a function of the amount of mussel tissue, basically the number of mussels in the sediment, and the phosphorus flux scales significantly with the presence of mussels. The cores that did not have mussels, so you could think about it, the sediment before the mussel invasion, plot all the way down here. The flux of phosphorus are significantly less from natural sediment than from the mussel-dominated sediment. And this changes everything. Basically, it means that the typical picture of phosphorus cycling that I described is not the one that's really important in these lakes. What's important is the stories we described in this recent paper that came out earlier just about a year ago. We used, in that paper, we used the mass balance model to simulate the dynamics of phosphorus. We did, however, use the model mainly as illustration. So I'm not going to go into the details of the model here, but I'll try to explain the story in simple terms. And for that, we'll need to look just at the numbers on the inventory of phosphorus and the fluxes of phosphorus. Lake Michigan is an example, but the same story can be done for Lake Huron and probably for Lake Ontario. So for Lake Michigan, phosphorus in the water column is on the order of 18 gigagrams. So we're looking at all of the phosphorus that's present inside the water column. Phosphorus that's contained in mussel tissues based on the amount of mussels is about half of that. So it's significant compared to all of phosphorus present in the water column. For fluxes, the external inputs are on the order of 4 gigagram per year. Similar amount of phosphorus settles down to the bottom of the lake. Mussels, on the other hand, uptakes through active filtration of the water column, so active filtration of particles, 36 gigagram per year. So 9 times higher than natural sedimentation. The benthic exchanges are greatly accelerated. They can also grow. So this is on the order of 1 gigagram per year. So in a couple of years, they can consume all of the annual external input. So they're significant contributors to phosphorus cycling. Here is another view of that. This is the diagram of what we think could be a representation of Lake Michigan before the invasion. Phosphorus gets into the lake. Most of it gets buried. A small fraction of it gets released. This is after the invasion. Instead of this small sedimentation flux, mussels sequester enormous amounts of phosphorus out of the water column. The lake mixes twice a year. And mussels have access basically to the entire water column during the times that they put it into the biomass, they excrete it. When they die, their bodies decompose. And so a lot of phosphorus goes in and a lot of phosphorus goes out. This is another view of this, of how the benthic fluxes may have changed as a function of time, right? So around year 2000, mussels invaded. Feeding was significant, quickly became a significant sink of phosphorus for the water column. Excretion, exhaustion balanced that. The net effect is this mussel net sink. It's a sink because the biomass was increasing, mussels were growing. For the net burial effect, there was a net burial effect, maybe not as strong, but the point is that the fluxes increased significantly. And increase in fluxes into and out of the benthic system means that the efficiency of recycling increased, because that's what it is. It's a percentage of phosphorus, of the settling phosphorus that gets recycled. So mussels greatly increase this percentage. So for Lakes Michigan and Huron, we were about here, about 20-25%. If you account for the fluxes of mussels, we're somewhere around there. So this means, from simple mass balance, this means that there is a time scale of response for the lake has greatly increased. If you change external inputs of phosphorus, the total phosphorus in the lake will respond with a significant time lag. Now it's on the order of several decades. We're losing sensitivity to external inputs. You can think about this as internal loading, and if you're used to thinking about managing of smaller lakes where internal loading is a problem, then you may be familiar with the problems that we may be facing now with these lakes. Again, because we increase the recycling efficiency, it's equivalent to these lakes having very high internal load. So, except that mussels are more unpredictable in this external loading because it's biology. Their populations fluctuate. But it may be a nice way of thinking about the lakes. It's analogous to internal loading. Okay, so what can we expect? Well, if the quagga populations are increasing, they will consume phosphorus until they starve. So this starvation limit is determined physiologically. We calculate it in the paper. You can look it up. But basically, phosphorus concentrations in the lake will decrease until the populations stabilize because they starve. If the quagga populations decrease, well, the effect will be decreasing, but now the time scale is really long. So the phosphorus in the water column will be coming back to its value that it would be without mussels, so externally imposed limit, but now slowly. In reality, quagga populations can fluctuate, they do fluctuate. So phosphorus, it can be proven pretty much mathematically out of the mass balance that phosphorus in the water column will be fluctuating, but still within these limits, and we can calculate these limits. So that's important because we can now predict the typical ranges of phosphorus for these lakes. So this is the simulation, this actually from the model for Lake Michigan, Lake Huron, Lake Ontario. Here is the same limits for each lake. And so we can specify these ranges within which we expect the total phosphorus concentration to remain in the coming years, even as quagga mussels populations fluctuate. Lake Erie is flushed a lot quicker. Its hydrological residence time is much smaller, and for this reason, these limits are very close together, and these lags that we're talking about, this lengthening of the time scale, is not as pronounced, which is very fortunate for Lake Erie. This means that the lake is likely to respond to changes in external phosphorus inputs. So if we want to decrease the productivity of the lake, decreasing external inputs should be still a viable strategy, at least from the mass balance point of view, at least from the fundamental constraints. So this brings me to my conclusion. I hope I'm not terribly over time. So I'll reiterate here. The phosphorus cycling regime is now controlled by quagga mussels. It's not so much by external inputs, except in Lake Erie. The responses to external loadings are going to be fairly slow, and changes in quagga mussels populations can have large effects on phosphorus supply. If they die suddenly, phosphorus concentrations can go up for a few years. If they start growing, phosphorus concentrations can collapse. So large phosphorus fluctuations driven by population dynamics, which is less predictable than typical chemical physical changes. Most of the biogeochemical cycling now happens through the sediment because mussels have access to the entire water column, at least for part of the year. The cycling of substances, well, organic carbon and nitrogen and phosphorus, now happens in the sediment. We don't see a lot of evidence that the properties of the sediment below a couple of centimeters are being modified. So most of it cycles in the topmost millimeters, plus of course the bodies of these quagga mussels. But this means that the biogeochemical cycling that used to be taking place in the water, high up in the water column, now takes place to a large degree at the very top of the sediment. And this may need to be taken into account. Processes like sedimentary suspension may be important and so on. Thank you for your time. I'll stop here.
Thank you so much, Sergei. That was wonderful. Any questions that anyone has for him, you can go ahead and type those in the chat, and then we will get to those after our second presentation. Okay, so next up we have Derek Schlea. Derek is a, oops, sorry, losing my screen. There we go. Derek is an ecological engineer working at LimnoTech's headquarters in Ann Arbor, Michigan. He has expertise that includes watershed and eutrophication modeling using various frameworks. Along with his colleagues and other collaborators, Derek has developed and applied models to water bodies throughout the Great Lakes region and to the nation in order to aid the planning process for solving complicated water quality issues. And then I will stop sharing, and Derek, you can take it away. Thanks so much.
All right, great. Just confirming, give a nod that you can see my
Yep, it all looks
slides.
good.
Okay, great. Well, yeah, good morning, and thank you for attending to everyone out there, as well as your interest in hearing me speak about the Lake Erie ecosystem model. So I'll be talking about our work really over a 2.5 to 3 year period in assessing ecosystem indicators for the western, central, and eastern basins of Lake Erie. This work was funded by the Great Lakes Restoration Initiative via the U.S. Army Corps of Engineers Buffalo District. So my goal for today is to cover a lot of ground, but at a relatively higher level, to give you an overview of the entirety of the project, beginning with a brief background on the history, motivation, and objectives for the work, and then moving into the model framework, capabilities, the model development process and calibration, and then wrapping up with a look at some of the diagnostic or exploratory scenarios that we did. So first, I want to give you a brief history of the major milestones in the Lake Erie world over roughly the last decade as it relates to the Great Lakes Water Quality Agreement, in particular, the Nutrients Annex, or Annex 4, which in 2012, the US and Canada committed to establishing binational phosphorus load reduction targets for Lake Erie as it relates to desirable ecosystem endpoints. And then the following year in 2013, a subcommittee and task teams were established. And eventually that led to some modeling work in 2014 and 15, where 9 different models of varying complexities and also varying elements that they were simulating as endpoints were used to inform phosphorus loading targets for the lake. And then in 2016, phosphorus loading targets were officially adopted for the western and central basins. Notably, no targets were adopted at that point for the eastern basin. And then lastly, in 2018, both at a federal level and state level, domestic action plans were developed that included descriptions of phosphorus load reduction measures that would need to take place in order to achieve those targets that were set a few years back. So really this leads us to the motivation for the LEAM that I'll be discussing today, or the Lake Erie ecosystem model. So as part of that multi-modeling work, or ensemble modeling work that I mentioned, there were several gaps identified, including the need to represent integrated water sediment and biological processes across the entire lake, as well as the need to represent all of the eutrophication response indicators, or ERIs, including cladophora, which is more pronounced in the eastern basin. So responsive to recommendations from a science advisory board convened by the US EPA, they recommended that WLEAM, or the Western Lake Erie Ecosystem Model, which is one of those 9 models used in the load target, the phosphorus load target setting, that it be deployed for the entire lake in order to provide better information on how to understand the load reduction impacts as it relates to both hypoxia development in the Central Basin, and also consideration for embedding a cladophora model within this eventual whole lake model. And that leads us to the objectives for this Lake Erie Ecosystem Model project, which started in late 2018 and then was completed in March of 2021 in an initial phase. So the objectives listed here were to develop a mass balance model to simulate the impact of pollutant sources on all of the ERIs. And again, to emphasize, that's harmful algal bloom production primarily in the western basin of Lake Erie, which is shown over here. I'm not sure if you can see my mouse or not. Hypoxia formation in the central basin, and then cladophora production in the eastern basin nearshore areas. And so it would also provide a tool for quantifying the impact on ERIs for various scenarios. And to emphasize, the intent or purpose of this was not for new ensemble modeling to end with a decision on revised phosphorus load targets. That was not the intent. Rather, to eventually develop a model to support the long-term adaptive management process for Lake Erie, and that could be one or more models as part of that. And so the application may include both diagnostic and scenarios, as well as forecasting uses. Okay, so that was a brief look at the background. Now I'll jump into the model framework and capabilities. So here we have a diagram depicting what the Lake Erie ecosystem model really is. So the framework consists of 3 separate models that are linked together via output from one model being fed into input as another model. And that consists of a hydrodynamic and thermal model called FBCOM, coupled with a wind wave model using a framework called SWAN. And then lastly, kind of the core model is the Advanced Aquatic Ecosystem Model, or A2EM, shown there in the middle. And so the way these kind of complicated modeling systems work is you have really a slew of inputs as depicted on the left. So that could be the atmospheric forcing functions, physical definition of the lake itself, including bathymetry and water levels, and then tributary and other loading from things like point sources. So that may be the flow, nutrients, and suspended solid loading. And then also defining inputs for the sediment bed condition, nutrient concentration, substrate type, and things like mussel densities. And then there's a variety of model outputs ranging from water quality state variables, phytoplankton or algae, and then really at the bottom there, the eutrophication response endpoints that are of primary concern for Lake Erie. So, stepping back, there's really a decades of research and science that's well established that serves as a foundation for a eutrophication model like LEAM or A2EM, and that consists of well-established relationships between nutrient availability, water temperature and clarity, and algal growth. Relevant to LEAM are 2 important biological submodels: the Great Lakes Cladophora model, which was recently updated in version 3 by Kaczynski et al., as well as the dreissenid mussel bioenergetics model, originally developed by Schneider and then updated by Bierman in 2005. And so in addition to that foundation of scientific research, the LEAM also draws on important data sets from the last 10 or even 15 years to support both the calibration and model development process. And that includes, research by various agencies or entities on monitoring things like nutrient concentrations, dissolved oxygen, chlorophyll levels, estimates of harmful algal bloom biomass from remote sensing-based techniques, and then also monitoring of dreissenid and cladophora densities on the bottom of the lake. So as I listed on that earlier slide, the output capabilities of A2EM include various water quality constituents that are listed here, including multiple forms of organic carbon and multiple species of both organic and inorganic nitrogen and phosphorus. There's also a fully coupled sediment diagenesis submodel, which simulates the diagenesis processes, including SOD generation and sediment water nutrient exchanges. And then the biological components of the lower food web are shown there on the right, with phytoplankton, zooplankton, cladophora, and dreissenid mussels all represented. This P or phosphorus cycling diagram depicts the physical, chemical, and biological exchanges or interactions that happen within the water column of the A2EM model. Starting at the top, the model again is driven by different forcing functions, including solar radiation, as well as external phosphorus or nutrient loading. And then in the green boxes, we have the different biological components represented, and once again, the various interactions that occur between the biological components, as well as some chemical and physical interactions that happen also between the water column and the sediment bed, including settling of particulates, free suspension, and that diffusive exchange. I'll dive into the dreissenid mussel submodel in particular for this audience to describe it a little bit. So the dreissenid mussel submodel is not a population model. It's a bioenergetic model, which means we have a fixed number of individuals across 3 different cohorts: a young of year cohort, a smaller or juvenile mussel cohort, and then a larger adult mussel cohort. And so with the bioenergetics model, it represents kind of what you see here in this diagram, the effects of mussel filtering on various water column constituents like phytoplankton, zooplankton, organic and inorganic solids, as well as the increase and decrease of the biomass of mussels themselves through either the growth processes or losses like excretion, respiration, reproduction, and digestion. And then once again, it interacts with the sediment bed and therefore the sediment diagenesis submodel as well. So jumping into a quick look at the model development process, this is a look at the simulation grid. So it's based on the LEOFS, or Lake Erie Operational Forecast System model that was originally developed by NOAA and other partners. It uses an unstructured grid, which gives you the flexibility of having more refined spatial grids or resolution in the nearshore areas, and then less refined or larger grids in offshore areas. So there's 6,306 nodes total and almost 12,000 elements, and that's also spread out over 20 vertical layers. One particular addition for the LEAM project was the Sandusky Bay, which is kind of a larger embayment of Lake Erie between the western and central basins on the southern southwestern area of the lake. A few of the initial conditions that we need to define, or actually stepping back with the bathymetry shown on the left. So we use the NOAA NCEI bathymetry data set for most of the lake, but then we had a separate data set for Sandusky Bay that defined its bathymetry. And for those of you not familiar with Lake Erie, the western basin, it's the smallest spatial basin, but it's also the shallowest. It's quite shallow, just a few meters deep, and a lot of the nearshore areas transitioning to 5 or 10 meters deep in some of the deeper portions of the western basin. And then we have the much larger central basin, which also gets quite a bit deeper than the western basin. But then we transition to the eastern basin, which is the deepest of the 3 basins, slightly smaller in spatial area than the central basin, before water flows out through the Niagara River to Lake Ontario. On the right, we have a look at the Lake Erie bottom substrate definitions. So based on a data set available from the Great Lakes Aquatic Habitat Framework shown there on the bottom, we defined for any given model grid or node a substrate type, either sand, mud, clay, or hard substrate, and that was useful for informing subsequent model input definitions like the sediment, phosphorus, nitrogen, organic carbon concentrations. So that was somewhat based on a function of the substrate, as well as some sampling data that were made available. And also substrate was used for the suitability for cladophora colonization. The next model input to describe is the lake bottom mussel density and distribution. So the inputs that the mussel submodel actually needs is a population density, the number of individuals per meter squared, and then also a mass-based density, so the grams of carbon per individual, and then we put that together. And what I show in this map is the mussel biomass density expressed in milligrams of carbon per meter squared. And really there were two primary data sets that informed this for Lake Erie. The one shown is published by Karatayev in 2017, Journal of Great Lakes Research, that studied mussels throughout the entire lake at the stations shown in this map during the 2014 CSMI year, special sampling year for Lake Erie. And there was also a data set from the Environment Canada's Great Lakes Restoration Initiative. I don't have that depicted, but that was also important for informing the initial mussel densities. So what we did is varied the mussel biomass based on this data set, really, which suggested that in the deepest areas of the eastern basin is where the highest densities resided, as well as overall the eastern basin had the highest density of mussels. Whereas the central basin, mussels were absent at a lot of the sampling stations or limited to very young of the year mussels. So, and it's largely driven by the annual hypoxia formation in the bottom areas of the central basin that prevent the establishment of mussels there. And then the western basin is really somewhere in between, not nearly as dense as the eastern basin, but not the absence like you see in the central basin. The last model input that I'll cover, and again, there's really dozens of more model inputs that go into LIM, but in the interest of time, focusing on some of the key ones. So this is a look at the tributary loading. In particular, I have a pie chart of the total phosphorus load by source location, and this is actually averaged over a 10-year period, 2008 through 2017. Where we have a look at some of the major tributaries or phosphorus loading sources. The Maumee River and the Detroit River together contribute over half of the phosphorus load, and then other Western Lake Erie Basin locations, when you add those in, that's almost 3/4 of the load come into waters of the Western Basin. So what we have depicted in this map are in green shaded areas are the watershed areas draining to the lake that are monitored by flow gauges by either USGS or Environment Canada gauging stations. And then in the orange areas are some of those unmonitored areas. So the point being here is that we represented the watershed loading for the entire loading draining to Lake Erie. We had, I think, 70 tributary sources represented coming into our model. And that took a mix of deciphering the observed data, both flow and nutrient concentrations at various sampling locations, and then coming up for some assumptions in these in-between areas that maybe weren't monitored for flow, maybe there were some nutrient concentrations. All right, so that was a kind of a snapshot of the model development process. We'll jump into the model calibration. And again, I'm going to cover this at a very high level in the interest of time, just give you a snapshot or higher level of the work that was involved. So now that we've developed this model, the primary calibration years were the 2014 through 2016 period, so 3 different years. 2014, as I mentioned earlier, was a CSMI year, so there was relatively more abundant sampling data that we could use to calibrate the model to. And then also in 2014, that's when you might be familiar, the Toledo water crisis happened in August of that year for a few days, when the city of Toledo had to tell residents not to drink water because of microcystin toxin detection in their drinking water. And anyway, as an outcome of that, it really amped up the monitoring efforts in 2015 and 2016 as well. So things like more buoys deployed, more stations being visited more frequently. So this 3-year period was relatively abundant data-wise, and that worked well for the model calibration. As noted here, we also simulated 3 other years as corroboration years. So the parameters we calibrated to are a variety of physical, chemical, and biological parameters, sampled by multiple agencies at different times. And that's really the primary water quality monitoring stations or programs are shown in this map in the bottom right, which were the Environment and Climate Change Canada sampling program, US EPA's GLMPO monitoring program, and then specific to the Western Basin of Lake Erie, sampling programs by NOAA GLERL and the University of Toledo. were very valuable in calibrating the model. Oops, sorry, I jumped ahead there. The strategy that we follow is called a weight of evidence approach, where rather than relying on any single measure to say whether the model is, acceptably or unacceptably calibrated, we use a variety of both visual and statistical measures in what's called a weight of the evidence approach. And it also relies on both hard and soft calibration targets, if you will. So the hard data are more or less the point in time, point in space samples like nutrients or dissolved oxygen or chlorophyll or whatnot that you can compare an exact model output against. But the softer targets, those are things that aren't necessarily measured in the same way, but there's maybe some institutional knowledge or scientific knowledge about what's happening. And that could be things like the internal phosphorus flux. Is the model representing our best understanding of the loading of phosphorus from the internal sediments? Also, are the mussel and cladophora growth curves behaving as we might expect from the best available literature? So those are the soft, almost process-based targets that we also check when we're calibrating the model to ensure it's behaving properly. Okay, so skimming through some of the results here. Again, in the interest of time, this will be a high-level snapshot of some of the types of comparisons that we do and the parameters that we look at. On the left plot, we have an example model data comparison of a daily time series of water temperature for the entire lake surface. So this is a spatially aggregated time series that's produced annually by GLSEA out of NOAA observational data set. And then we compared the LIM simulated in the dark blue line against that. On the bottom right, we have a bar chart comparison of the average simulated and observed total phosphorus concentrations over that 3-year period, split out for, I think, over 50 different sampling stations for those 4 primary sampling programs that I mentioned. And here we're seeing that the simulated values are matching the trends in the observed values really well, with the highest concentrations in nearshore areas of the Western Basin transitioning to the lowest concentrations of phosphorus on the eastern side of the lake. Next, we have a look at a daily time series again, but this is for an individual location in the Central Basin that was monitored with continuous DO measurements in the bottom layers to get a feel for the hypoxia. So this dissolved oxygen plot shows that the model is reasonably well reproducing the temporal patterns in dissolved oxygen concentration. So we're not always, hitting those data points perfectly, but overall we're matching the trend of when the decline is happening. And also we're looking for really the number of hypoxic or anoxic days. So is the model capturing the number of days that DO drops below 4 or 2 milligrams per liter? And also, is it capturing the trends annually? So in this particular station, 2016, really had the worst hypoxia, or the most number of days below 4 or 2 milligrams per liter, followed by 2015, and then 2014 had the least, and really the model captured that trend quite well. Stepping back to nutrients on the bottom right, we have a plot of nitrate concentrations now. So this, again, is an average simulated versus average observed for the different stations. And again, showing that the model is matching the spatial trends quite well for nitrate. Okay, lastly, a few additional comparisons. On the left, we have a map-based comparison of chlorophyll concentration simulated by the model compared against a remote sensing based product of surface chlorophyll concentrations on the left-hand side. And this is shown for roughly the peak time of phytoplankton or harmful algal bloom growth in each of the 3 years simulated, 2014, 15, and 16. And overall pleased with how the model is matching both the relative magnitude of those chlorophyll concentrations between these 3 years, as well as some of the spatial trends. And then lastly, on the bottom right, so this is looking at that full 6-year period that I was talking about, comparing the model prediction of the seasonal HAB severity, which is quantified as a 30-day average biomass in metric tons. That's kind of the agreed upon metric for quantifying the annual HAB severity. So that's compared against 3 different independent remote sensing type estimates of the HAB severity that was recently published in a paper. So overall here, once again, the model's capturing that annual variability across these 6 years, with 2015 being the worst bloom, 12 and 16 being relatively mild blooms, and then 13 and 17 were also rather significant blooms. So the model's matching those trends across the 6 years relatively well, and also the magnitude in terms of the biomass. Okay, so again, high-level overview of the calibration process, but want to move on to the diagnostic or exploratory scenarios to show some of the utility of this model. So now that we have developed and calibrated this sophisticated model that's built on this foundation of scientific principles and research, we put it to good use or apply it by running various hypothetical scenarios. So to do this, we modify the model inputs shown on the left, run the model or models if we need to run multiple models, and then evaluate the outputs shown on the right to understand the responses and causes and things like that. And this matrix illustrates the type of eutrophication response indicators, or ERIs, that we evaluated during this 2-year project with LIM. And really, it's one of the benefits of a process-based model is that there's somewhat limitless possibilities to evaluate the model output and responses in both space and time. So for these 9 different metrics shown here, we had different spatial averaging techniques, different temporal averaging periods, and even different vertical averaging depths, as well as different aggregation types that all met the need at what we were trying to evaluate with the LIM in terms of the ERI and how it might have been defined as part of the Great Lakes Water Quality Agreement. And again, to emphasize, one of the utilities of this model is that ability to diagnose things like the mass balance exports and understand the why, and explain potentially counterintuitive outcomes as well. Why is the model suggesting that this is the response we're seeing? So although we ran a variety of scenarios, including hypothetical external load reduction scenarios as part of our project, today I want to focus on just 3 diagnostic scenarios that we simulated. And these were done to elucidate the relationship between mussel-mediated processes as well as that sediment water nutrient exchanges. So we did a 50% reduction in the initial mussel biomass as a hypothetical scenario. Another one, we turned off that sediment water nutrient exchange capability in the model, so we removed those nitrogen and phosphorus fluxes. And then the third scenario combined the effects of those 2 items. So to emphasize here, these are not suggested policies necessarily, nor feasible actions that can be taken, but really they're meant to help us understand the importance of the many drivers in this complex ecosystem that is Lake Erie. So just a snapshot of the types of results that we produced. This plot represents a daily time series average for all of the Western Basin model nodes, and it's for 2014 only. So we did only run these scenarios for 2014 because it was somewhat of an intermediate year in terms of harmful algal bloom production. And this is a look at the average chlorophyll A concentrations over the March through October period. Baseline is shown in black. That decreased mussel biomass is shown in blue. The turning off the sediment phosphorus flux is shown in green, and then the combination is in orange. So as you can see, with all of these scenarios, there was at the peak anyway a slight reduction in that chlorophyll concentration with the decreasing the mussel biomass, but a more pronounced reduction by turning off that sediment phosphorus flux. It was, I think, roughly a 10% reduction in the peak harmful algal bloom with that no sediment phosphorus flux. And then when you combine those 2, there's a nominal decrease as well in the chlorophyll concentration. So jumping to the Eastern Basin, Cladophora, on the other hand, responded somewhat differently. It was significantly impacted by the reduction in biomass, mussel biomass, but not by the elimination of sediment nutrient fluxes. So based on these results, the presence of mussels in that baseline at double the biomass really facilitated the growth of Cladophora by both clearing the water column of suspended materials and thereby increasing the light availability for Cladophora growth, as well as increasing the availability of dissolved phosphorus in bottom water layers. And it also shows that, at least in eastern basin, according to our model, that sediment phosphorus flux was not a significant contributor to fueling cladophora growth. Okay, so again, kind of a rapid fire look at the scenarios there, but really the key findings from these diagnostic scenarios were that the model results were generally consistent with expectations. As I mentioned, the elimination of the sediment phosphorus flux reduced HAB severity in the Western Basin by about 10%, but it was minimally impacted by the reduction in mussels. Hypoxia, I didn't show any results there, but really the individual scenarios as well as the combined scenarios had minimal impact, less than 3% change in the hypoxia metrics that we quantified. And for cladophora, it was a bit of a confirmation that the presence of invasive mussels was beneficial to cladophora production, because when we reduced the biomass in half, we saw about a 20% hit or reduction in the cladophora biomass, whereas minimal impact by eliminating the sediment phosphorus flux. So to wrap up here, I've just went over a higher level overview of the Lake Erie ecosystem model development, calibration, and application project. We simulated the 6-year period, 2012 through 2017, represented loading for the entire watershed, nutrient loading from the entire watershed sources, as well as point sources and atmospheric sources. And then calibrated the model to represent all of the eutrophication response indicators of interest, really accomplishing the objectives that we set out to do so. There is ongoing work currently in the second phase where we are extending and evaluating the model to simulate the 2019 and 2020 periods. And we're also planning on linking the model to one or more watershed models so that the flows and nutrient loading from the watershed models could be coupled with the lake model. Some potential future directions that we may explore, just throwing out here, are representing complete water years. So actually starting the model on October 1 and running it over the winter, and then revisiting the calibration to see how it holds if we represented that full water year, as well as simulating the long-term evolution of the sediment bed, really relevant to the previous talk. We're only doing seasonal simulations here right now with LIM, so it could be valuable to apply the model in a longer-term setting to see how that sediment bed evolves, especially in relation to changes in external loading, for example. Yeah, so that was it. Thank you for bearing with me there, and I guess I'll turn it back over to Sam and we'll field any questions.
Great, thank you so much. Ken is going to lead our Q&A. So if you do have any questions, please type them into the chat box, and I will just share our presenters' contact information if you should have any additional questions that you'd like to follow up with them on.
Yeah, great. And thank you, Derek and Sergei, for your presentations. The first question may have been answered during Derek's presentation, but I'd still like to ask. It was posed during Sergei's presentation, is why isn't there information on mussels for Lake Erie?
Yeah, of course, there is information as Derek showed, and the surveys that were done by Alex Karatayev's group in particular have documented them quite well. I did not show those data in my talk because we haven't worked on Lake Erie. We were recovering sediment cores, looking at a smaller number of locations in more detail. So I just didn't present those data because they were not my data.
Sure, great. Next question, I think too, was during your talk, Sergei, was how does one determine if our inland lake sediment porosity will support quagga mussels?
Well, in our data, we rarely see mussels in sediments that are below 90%, 88, 89% porosity. I'm not sure if this can be transferable, depending on how similar mussels are. I'm guessing to some degree this may be transferable. So if the sediment is soft and with porosity above 0.9, 90% deeper than a couple of centimeters. I'm guessing it would be less likely to support quagga mussels, but that's a guess.
Sure. Again, during your talk, Sergei, as the P levels decrease to the mussel starvation limit, how is that expected to affect other life forms in lakes, such as the native mussel species?
All right. Again, a biologist can probably answer this question better than I can. So the effect of phosphorus in mussels is really through the food, through phytoplankton. I'm not. I don't really know the physiological limits for the native mussels. I'm guessing they are not as tolerant as quagga mussels to starvation. Quagga mussels, as Ted Ozerski's group has shown, they can starve for several months without noticeable effects. But I'm guessing quagga mussels are more tolerant, but I'm not really a biologist to answer this question.
Sure. All right, a bit of a longer one here for you, Sergei. One viewer was surprised by the difference in P uptake by mussels and sedimentation rates. Most of the mussel population is in deep water and dependent on sedimented material. Perhaps location of mussels becomes really important. Mussel biomass increasing in deep water may be less important, the biomass increasing in shallower depth. And they were wondering if you had any comments on that.
Yeah, that's a great comment. So yes, when we calculate the numbers that I showed in presentations were for the lake-wide population. And in the model, we calculate things based on mussel biomass. So there is a certain filtration rate associated with the mussel biomass. So because the deeper sediments have less biomass, there are definitely fewer mussels there. And they tend to be smaller. They filter at smaller rates. They consume, so they recycle less phosphorus. So for the numbers, for the lake-wide numbers that I've shown, the nearshore populations must have disproportionate contributions. So I think you're right that mussel biomass increasing in deep water may be less important than the biomass increasing in shallower depths. The dynamics near shore and offshore may be somewhat different because mussels have been established near shore for quite a while now, and they relatively recently expanded into the deeper waters. So the relative changes in the population may be smaller near shore. But I think because of the larger biomass near shore, their dynamics should be much more important.
Great. I think last one for you, Sergei, before we switch over to Derek. Any plans in your team for an ecosystem-wide assessment of changes of N and C?
Yes, we're working on it. It's somewhat more difficult to do for nitrogen and carbon what we did for phosphorus, because like nitrogen is not a productivity limiting nutrient. But we're putting together an analysis of the changes in the carbon cycle and nitrogen cycle, and hopefully constraining the carbon budget. At the moment, the preliminary data seem to suggest that the effects are the mussels don't modify the sediment geochemistry enough to cause significant changes to the recycling rates of nitrogen and carbon, but we're working on it.
Great, thank you. I think the next question here is for Derek, and it was question, does the model assume P limitation as opposed to N limitation throughout the HAB period?
Yeah, so I think the best way to answer that, it will simulate both phosphorus and nitrogen limitations. Yes, depending on the concentrations of the available nutrients as simulated by the model. So it does have that capability. Yep.
Great. Just a reminder to anyone, if there's any more questions, to enter those through the chat. We had a question of if the speakers were going to share their slides. We're going to share the recording for this talk, but not the slides. And I think we'll give it just a few more moments to see if there's any last questions. Derek, do you mind showing your conclusion slide again? We had a question for that.
Yeah, let me pull it back up here a bit. Excuse me. Oops. Okay, I think I.
And sorry, I'm seeing in the chat, did I not ask Hans' question about if the model assumes P limitation throughout the HAB period as opposed to N limitation? Or perhaps there was another question that I didn't receive in the chat.
And I guess I could elaborate on that
Sure.
answer. So yeah, again, the way the model works is it assumes, you know, the most limiting nutrient. So there's not, I guess, a co-limitation. So that there could be periods of, you know, either phosphorus limitation or nitrogen limitation in the model. You know, I don't have exact numbers or, you know, areas in space and time with, you know, whether or when that nitrogen limitation would or might occur during the HAB growing season. You know, I think by and large, we were seeing a phosphorus limitation as you might expect, but that's not to say that there could be certain areas of the lake where nitrate or nitrogen concentrations were much lower at times, and therefore was, you know, suggesting a nitrogen limitation. But I don't have those numbers offhand, just confirming that it can and does simulate both P and N limitations.
Sure, and thank you for that, Derek. So I think we've answered all the questions, and we can now take a moment to thank our speakers. And I think we have a closing slide with some contact information, as well as information for learning more about the HABs Collaborative and Invasive Mussels Collaborative. A recording of this webinar will probably be posted in the next week or so and available for you to view. So thank you again, Derek and Sergei, for presenting today. This was great. And thank you for everyone for joining us.